Hi guys, today we're going to be looking at a genetic modification question. Genetic modification of crops. Genetic modification of crops began with the discovery that the soil bacterium, Argobacterium, could be used to transfer useful genes from unrelated species into plants. The gene, called Bt, which produces a pesticide toxin that is harmless to humans but is capable of killing insect pests, is one of the genes most commonly inserted into crop plants. Many new GM crops such as maize, potatoes, and tomatoes are modified to be pest, disease, or weed killer resistant. GM foods could have unforeseen effects. Toxic proteins may be produced, or antibiotic resistance genes may be transferred to human gut bacteria. Modified crops could become weed killer resistant, superweeds, and modified crops could accidentally breed with other wild plants or other crops. Question 3.1.1. According to the passage, how did genetic modification of crops begin? So, as we can see in the passage, it began with the discovery that the soil from bacterium Argobacterium could be used to transfer useful genes from unrelated species into plants. Question 3.1.2. Explain why a plant which is modified to be weed killer resistant could be a problem for farmers. So, the modified crops could be a problem for farmers as they become superweeds, therefore being weed killer resistant. They also, and due to them being superweeds, it becomes very difficult to kill them or expensive to kill them. And as a result, it could outcompete the original crops that are growing in the area. Question 3.1.3. Give 2 examples in the passage of the use of GM crops that may be a potential threat to human health. So, as we see in the passage, they say that toxic proteins may be produced or antibiotic resistant genes may be transferred to the human gut bacteria. And that's all for this question. Thank you.